Hi, my name is Andrea Perez, and I will be presenting my project over genome editing using CRISPR-Cas9 to study ribosome specialization in Leishmania parasites. So Leishmania is a parasite that causes leishmaniasis, a tropical disease that affects around 12 million people worldwide. Visceral leishmaniasis is the most severe form of the disease, and it has almost 100% death rate. This happens when you ingest the parasite. There is still not a secure vaccine or an efficient drug available in the market. The way that this parasite works is that it is found in mosquitoes, and whenever mosquitoes bite mammals, then you get infected with it. So whenever the parasite switches from the insect to a warm-blooded mammal, it is exposed to different dramatic stresses, which include a different immune response, temperature rise, change in pH, and in nutrition. One of the molecular mechanisms regulating the gene expression during the host vertebrate infection are still very poorly understood, and around 60% of the Leishmania genome encode hypothetical proteins with unknown functions. So our hypothesis was that the ribosome, which is an organelle found in cells that is responsible for producing proteins for the cell to survive, undergoes a change in the composition during heat shock to support selective translation of stress response transcripts and parasite survival during the change of host. So this means that the ribosomes will undergo a change in translation and transcription in order to overproduce or underproduce certain proteins to help the parasite survive during this change of temperature. This is really significant because if we are able to identify the different transcripts and translational regulations during the heat shock, we would be able to develop better strategies and developing new medicine, vaccine, antibiotic in order to treat this disease and save thousands or even millions of lives. The experimental design entails first heat shock. So we grew cells at 27 degrees Celsius to mimic the temperature of a mosquito. And then we grew them at 37 degrees Celsius to mimic the temperature of a mammal for 2 hours. After that, we did a polysomal profiling. This just means that we look at the ribosomes present at this point, and we look at how fast or how efficiently they are producing proteins. Later, we looked at the proteins and we see the changes in the production at both the control and the heat shock. Later, we looked at one of these gene candidates called L36, and we genetically edited with CRISPR-Cas9 technologies in order to know the gene function. So here in this graph, you can see the heat shock induces a dramatic change in Leishmania major polysomal profiling. So the way that ribosomes work in Leishmania is that they can be in a pre-polysome form, a light polysome form, and a heavy polysome form. Heavy polysome meaning a lot of proteins are being produced really fast at that rate. So here the orange line is the heat shock and the blue line is the control. So you can see how the orange line is way higher at the pre-polysome, but is lower at the light polysomes and the heavy polysomes. When you look at the control, which is the blue line, you can see how the pre-polysome is lower, but the light polysome and the heavy polysomes are way higher. All of these when it changes from a mosquito to a mammal. So then we looked at some of these proteins. Heat shock protein 83 and heat shock protein 70 were both way higher at the heat shock response. So the orange bars, as you can see, at all levels. So both the three pre-polysomes, light polysome, and heavy polysomes were way higher during the heat shock response. However, when we looked at SMT and C14, which are non-heat shock related proteins, they're just normal proteins that the cell needs, were way higher for the most part at the control level, the control being the 26 degrees. Also, we did a proteomic analysis that revealed the dramatic change in monosome composition during heat shock. So this just confirms our data. Here, the blue lines represent the amount of ribosomes present at this point, and at this specific location is the monosomes. So you can see how at 27 degrees, the lines are lighter, and at 37, they're really heavy, which just confirms that monosomes are heavily involved during the heat shock response. So we are led to believe that they are really necessary for the survival of the parasite. And this for biologists is really important because We usually believe that the ribosome is a very standard conservative mechanism that just comes, produces proteins, and that's it. However, when we look at this data, it's incredible because we have found in Leishmania cells how the ribosomes change its composition. Literally, they become monosomes and they start producing and underproducing certain proteins. So here you can see how at 37 degrees, 6 of the proteins are being produced that are not being produced before. These are brand new proteins that we just hadn't known about. So we were really interested in knowing what these proteins did that helped the parasite survive, or why were they only found in monosomes. So that is when the CRISPR-Cas9 technologies come. CRISPR-Cas9 is a technology that was discovered in 2015, and it just won the Nobel Prize in 2020. So this is a really new, innovative technology, and we were the first lab to develop the CRISPR technologies. We actually made it work for us, and it's a really easy and innovative technique that just helps us discover these type of things. So the way that it works is we infect some cells with CRISPR-Cas9 technologies, and these allow us to cut and paste parts of DNA. So some genes, we are able to either introduce some genes or take out some genes completely. Here, as you can see, the wild type represents normal cells without CRISPR-Cas9 technologies. So we tried to tag an L36 gene into a Leishmania cell at 26 degrees, the standard cell. You can see how the wild type has nothing, has no genes, has no L36 gene production. And when we tag it with CRISPR technologies, we genetically modify it, so this shows up, this L36 gene. We also introduce an M-neon green tag that allows us to use fluorescent light to see which cells have been infected. So you can see here the wild type, the blue laser shows nothing, but whenever you use the genetically modified cells, they shine green, which means that our CRISPR-Cas9 technology worked perfectly fine. Then we were interested in seeing what L36 did for the cell. So we know that L36 is a ribosomal protein, even in humans. So we were really, really interested in knowing what this did. Like, we could have this protein in common with the parasite. So here in this specific gel, you can see how the wild type has the gene, but with CRISPR technologies, we were able to knock out this gene, just completely erase it from the genome. And you can see here that it's gone, completely gone. Later, we also added an antibiotic resistance gene because this helps us select which cells are genetically modified as well. So here you can see how the knockout has the tag. We tagged antibiotic resistance, and the control, which is the wild type, doesn't have it. This is just to confirm that the CRISPR technology worked fine. The UTR junction just shows us where exactly do the genes align. And the SMT-ORF is a normal gene that let us see how serves as a control, basically. So you can see how the knockout and the wild type do still have these genes. We didn't erase anything else that wasn't the L36 gene. So then we grew the cells. So we had 4 different sets of cells that we grew. Here in the blue line, you can see how the wild type and the knockout grew at 26 degrees Celsius, and they grew perfectly fine. They exponentially grew, which would mean that if this was happening in a human, they would keep growing, reproducing, and keep infecting the person. However, when you look at the wild type at 37 degrees, you see how it grew less than at 26 degrees, but it was still, growing, reproducing, etc. But when you look at the knockout at 37 degrees Celsius, so our genetically mutant, let's say, it barely was able to survive. So you can see this is a really, really low level of cells. Even after 12, 30, 48 hours, the cell was not able to reproduce without this specific protein. So this leads us to believe that CRISPR technologies could cure essentially Leishmaniasis someday. Our results presented here support the statement that under a stress condition, the parasite is able to modulate the ribosomal composition. So the ribosome specialization occurs especially during heat shock. And proteins such as L36 participate in this. So this is overrepresented in monosomes, and is required for the proliferation of the cells, which eventually lead to the cycle of reproduction of Leishmania. So without this protein, Leishmania is not able to survive. We currently have unfinished data on some mice. Whenever we infected mice with this specific strain of a knockout, the parasites were not able to survive or reproduce, so it really affected their virulence. This is incredibly important because this could actually lead to saving thousands of people's lives. If we were able to somehow knock out this one protein from cells, the cells would not be able to reproduce, and eventually they would die with the use of certain antibiotics because they would be so weak they wouldn't survive. So the next step is to find out what the other 5 proteins do, keep testing, and hopefully someday we'll be able to find a cure for leishmaniasis and save millions of lives. And that's my project. Thank you.